Immunotherapy trains the body's own immune system to hunt and destroy cancer. But for many patients, this process eventually hits a wall, causing treatment resistance. To understand this failure, we have to look at the immune system's primary reconnaissance units, conventional type 1 dendritic cells, or CDC1s. These cells swallow fragments of the tumor and display them on their surface as targets. This acts as a molecular flag, alerting the cytotoxic CD8-positive T cells exactly where to strike. Entering a tumor is a move into a harsh, shadowy environment. This nutrient-starved wasteland places extreme metabolic stress on cells trying to operate within it. Under this pressure, the dendritic cells lose their functional fitness. They begin to drop the tumor signal, leaving the killer T cells dormant and unable to find their targets. Immunotherapy often fails not because the T cells are incapable, but because the messenger cells are too starved to deliver the coordinates. Researchers found that exhausted dendritic cells inside tumors aren't all the same. They actually separate into two distinct groups based on the health of their mitochondria. This flow cytometry plot identifies the split. The top left cluster represents cells with polarized, healthy mitochondria, while the larger cluster on the right shows cells with depolarized mitochondria that are beginning to fail. In this electron microscopy image, the healthy mitochondria display sharp, tightly stacked folds called cristae. These structures are the physical requirement for a cell to produce energy. Researchers identified the mitochondrial fusion protein OPA1 as the specific factor that determines which dendritic cells stay functional and which ones collapse. OPA1 activates a signaling pathway through a protein called NRF1. This maintains the cell's oxidative phosphorylation, keeping its energy engine running at full power. When OPA1 drops, energy production halts. The dendritic cell is cut off from its power supply and enters starvation. In a desperate attempt to survive, the cell triggers autophagy, a process where it begins to digest its own internal parts to find fuel. This diagram shows how that survival tactic backfires. Without energy, the cell inadvertently breaks down its own MHC1 molecules and tumor antigens. MHC1 is the physical platform to present targets. When digested, the signal is blocked, leaving the immune system blind to the cancer. The loss of a single mitochondrial protein forces the dendritic cell to consume its own messages, effectively hiding the tumor from the body's defenses. This led researchers to a hypothesis. What if we could ensure these dendritic cells were metabolically charged before they even encountered the tumor? To test this, they isolated dendritic cells with healthy OPA1 active mitochondria and injected them directly into tumors in mice. These line graphs track tumor volume over time. While tumors in the control groups grew rapidly, the lower lines show how the energized dendritic cells achieved significantly better control over the cancer. The researchers then combined these recharged dendritic cells with existing checkpoint inhibitors, which help the immune system maintain its attack. The results were dramatic. A significant number of mice not only rejected their tumors entirely, but developed a long-term immune memory that protected them from future cancer challenges. Durable immunity requires a metabolic infrastructure that can survive the battlefield. By fortifying the cellular power supply, we ensure the immune system's messengers never lose the ability to communicate their target.